Hi, welcome to your virtual classroom and the lecture for Chapter 6, which is the start of Unit 3, the endocrine system. I'd like to first review with you the learning goals for this system. Major endocrine organs, which were covered in Homework 6, are listed here: the pituitary, the adrenal glands, thyroid and parathyroid glands. We'll also need to review chemical classifications of hormones, steroid hormones versus peptide hormones, and water-soluble versus fat-soluble. That will help you understand the hormonal actions and interactions with target tissues. One key feature here is the use of the circulatory system for delivery of these essential chemical ligands to every tissue in the body. The endocrine system is the body's second great controller, behind the nervous system, which we just studied in Unit 2. This is going to be achieved by chemical hormones or ligands that use the bloodstream for access to all body tissues. Your primary endocrine organs are going to be the hypothalamus, pituitary, thyroid, parathyroid, adrenal, pineal, and thymus. Those are all detailed here in these class notes, and I will go through them with you now. This is a 2-lecture series. We'll try to get through all of this information for Chapter 6 in one shot. Other tissues that we'll discuss at the end of this lecture will be secondary, normally because they're thought of as having other functions besides hormone function. So adipose tissue, the heart, kidneys, stomach, small intestine, they typically do other jobs for us, but they do also secrete hormones, and I'll list those at the end of this lecture. So endocrine organs are ductless, and they secrete their hormone products into the bloodstream. And hormones have these long-distance chemical signals that allow for cell-to-cell communication. Hormones are chemical substances secreted into the ECF or interstitial fluid and use the bloodstream to access all body tissues and regulate whole body metabolism, or BMR, which we measured in Lab 6. I will try to emphasize the experiments that you conducted in Lab 6 and expect lecture questions and exam questions to also emphasize the extra practice you all received by doing a laboratory on hormones that use the hypothalamus pituitary connection and the thyroid gland, which secretes T3, T4, which regulated basal metabolic rate in our rats. The characteristics of these long-distance chemical communicating ligands is that they tend to have prolonged effects once they arrive at the target tissues, and there's a lag time, which can range from minutes to hours, and in some cases weeks, before they get into therapeutic levels in the bloodstream. Our classifications for hormones are going to be neuro, which we'll start with those since we just finished the neuro Unit 2, amino acid-based hormones, or steroidal hormones. So let's start looking at the major endocrine organs in our notes. This should look familiar from Homework 6. And you were asked to classify the hormones and associate the hormones that come from the particular glands that are listed. And then you were to list the secondary endocrine organs and their hormones on the right of this diagram. Hormones produce one or more of their following characteristic changes in target cells, such as altering plasma membrane permeability, stimulating protein synthesis, activation or deactivation of enzyme systems, and they induce secretory activity, including stimulation of mitotic cell division. These types of activities are major if you're a cell. So hormones regulate a lot of cellular activities, and that's why they are so powerful and is an important topic to understand as we're managing clinical patients. So let's start with the neurohormonal axis, the connection between the hypothalamus and the pituitary gland. The hypothalamus sets hunger, thirst, body temperature, and it has both neural function and hormonal function. And its hormonal function is going to be targeting the pituitary gland. The pituitary gland is connected physically and electrically to the hypothalamus through the infundibulum. This forms a link between the nervous system and the endocrine system, and this controls metabolic activity that's under autonomic control. The pituitary gland, shown here in figures 6.2, 6.3, and 6.4 in your assigned textbook, is a bilobe central nervous system structure that secretes 8 major hormones. The anterior portion, or the forward portion, is true endocrine glandular tissue, and it's responsible for the synthesis of 6 different hormones, while the posterior portion, highlighted here, has long neural axons extending from the hypothalamus down into the posterior pituitary, and that is true neural tissue. We will see dual function in a lot of these glands where they have neural activity and they also have true glandular activity. So this is just one example, and we'll see many others when we study the glands individually. So the neural tissue or neurophysis secretes 2 peptide hormones under the control of neural stimulation from the hypothalamus, and those hormones are oxytocin. We first used oxytocin as an example of positive feedback control in Unit 1, and that hormone is around during labor and delivery. And antidiuretic hormone, ADH, or vasopressin, is a hormone we'll come back to in Unit 4 when we start learning about the kidneys. Both hormones are synthesized in the soma of the neurons in the hypothalamus, and they are sent along to the posterior pituitary through these long axons extending down through the infundibulum and accessing the posterior portion of the pituitary. Now, if we switch to the anterior portion of the pituitary gland, this is true glandular tissue. Notice that there are no neural axons extending down into this portion of the gland. All you see is capillary blood flow. So the cells in the hypothalamus secrete trophic hormones that communicate with the anterior portion of the pituitary. So trophic hormones, by definition, are hormones that regulate the release of other hormones. So they usually form this domino effect or this cascading effect where chemical communication will stimulate other hormones. We also studied this in lab number 6, where you learned about thyroid releasing hormone, thyroid stimulating hormone, and then ultimately the release of T3 and T4 into the peripheral bloodstream. So the difference between neurotransmitters and hormones is transport via the bloodstream, and hormones give more diversity of effects on target tissues. Targets for both neurotransmitters and hormones must have specific receptor proteins present in order for the cell or tissue to respond, and there also has to be a way of inactivating the signal. So target cell activation depends on 3 factors: rate of secretion, or the blood concentration levels of that hormone, which takes time to achieve, the relative number of receptors on the target cell or the target tissues, and the affinity of those receptors for the hormone. Affinity simply means how likely are we to have an attraction and binding in that receptor. Okay, so here in figure 5, we're looking at the hypothalamus, highlighted in pink, the pituitary gland in the middle, highlighted in tan, and then the target tissue along the bottom in sort of a salmon color. So I've listed for you the 5 different trophic hormones. Remember, those are hormones that beget other hormones, and some vocabulary terms which you must be able to memorize or recognize in a question and not get confused. So I highly suggest you make a list, write these things out. Don't just simply scroll through or think that you're going to remember them from listening to a lecture. You need to make a list and get these terms highlighted in green organized in your mind, so when you get questions on the exam, you're not confused and you can pick the right answer. So the 5 different trophic hormones from the hypothalamus communicating to the pituitary gland and targeting tissues all over the body are classified as somatotrophs. Somatotrophs have to do with skin, bone, and muscle, and that hormone is growth hormone, or GH. Corticotrophs release adrenocorticotrophic hormone, or ACTH, which targets the adrenal glands, and we will look at this hormone again when we get to the adrenal glands in this lecture. Lactotrophs are concerned with lactation. They release prolactin. Thyrotrophs release thyroid-stimulating hormone from the pituitary gland. And gonadotrophs release follicle-stimulating hormone and luteinizing hormone. Both are present in men and women in different concentrations. So in this diagram, you have either a positive or negative stimulatory pathway indicated with the plus or the minus signs. And the shorthand version of some of these big named hormones in the hypothalamic section, PRH, TRH, GHRH, so those are gonadotrophic hormones. And you can follow the pathway with the arrows down to the target tissue. There's another version of this same information. This is not in your book, but I thought to put it in context of physiology and anatomy, the same information is shown here. So you can see the hypothalamus up at the top is where we kick off this cascade through the infundibulum to the anterior pituitary, and you can follow the 6 different hormones that maintain the target tissue's basal metabolic rate, their size, and production of their hormones. So from the anterior pituitary, we see prolactin targeting the mammary glands. We see growth hormone targeting its target tissues for growth. Growth hormone is important before puberty because it will extend long bones, but after puberty, the epiphyseal plates are closed, so it would only target muscle and fat. Many athletes use growth hormone to supplement their athletic abilities. TSH is targeting the thyroid. We'll spend some time talking about the thyroid gland to kind of review what we did in Lab 6. Adrenocorticotrophic hormone, or ACTH, is targeting the upper layers or true glandular tissue of the adrenal glands. We've already talked about the middle medullary layer in the Unit 2 neural, part of the fight or flight response where we release adrenaline into the bloodstream. So we'll review that. And then the gonadotrophs, which would be FSH and LH, targeting the reproductive organs, testes, and ovaries. All hormones are tightly regulated through negative feedback control. So in this diagram, we can assign the laboratory experiment where we studied TRH, TSH, and T3, T4 in this pathway. And be ready for this type of diagram to come up on quizzes and exams, and I can simply change out the endocrine gland. I can call for the adrenals or the pancreas or some other gland, and then you have to go backwards and talk about which trophic hormones regulate the release of the glandular hormone that targets the cell response. So be ready for using figure 6.5 and applying this control feedback loop for any of the trophic hormones that were assigned from figure 6.5. This is a way of regulating higher or lower hormone concentration levels in the blood. Okay, so now we can start talking about our major endocrine glands that were on our learning goals list on the second slide, and we'll have a section for each gland, starting with the thyroid gland, studied in laboratory number 6 with the rats. Here's a look at the thyroid gland. It's fairly superficial, and it's just distal to the larynx or the voice box. You can see the trachea extending down, and this butterfly-shaped thyroid gland just kind of hugs the trachea, which is shown in blue. There's two lateral lobes, left and right, and this gland will secrete T3, which is triiodothyronine, and T4, which is tetraiodothyronine. Iodine is a dietary requirement for the manufacture of T3, T4 hormones. So I'll show you in just a minute how iodine from your diet is incorporated into these molecules. And we've already seen with our rat experiments that the basal metabolic rate can be affected by high levels or low levels of T3 and T4. The synthesis of T3 and T4 occurs in follicles that produce a glycoprotein, which is thyroglobulin. And if we look at our histology, this is where you need to jump over to chapter 21. Part of chapter 6 assignment straddles chapter 21 as well, where we can look at the histology or the microscopic view of the thyroid gland showing these follicles. They're filled with colloid, and this is where T3, T4 is synthesized inside these colloid follicles, where you have thyroglobulin and dietary iodine incorporated together to make triiodo and tetraiodothyronine. In this slide from figure 17 in chapter 21, we can follow the pathway where iodine is incorporated into the follicle from dietary sources like table salt. And the peroxidase enzyme is going to combine iodine with thyroglobulin. And there's two forms or two precursors which act like building blocks for T3, T4. Monoiodotyrosine and diiodotyrosine. So one is mono, di meaning two. So if I add 1 plus 2, I get 3, which is triiodothyronine. If I take two diiodotyrosines, which are 2 plus 2 is giving you tetraiodothyronine, or the T4. So these ratios are found in peripheral blood samples. And TSH is going to stimulate the hydrolysis of T3, T4 from thyroglobulin and then initiate secretion out into the plasma, where T3, T4 will be stabilized by carrier proteins that are normally found in plasma and help them arrive at nearly every cell in the body. This is just another figure of how the exocytosis, or the secretion of T3, T4 occurs, and it has access to the bloodstream. Here's a look at T3, T4. Okay, so T4 is on top, and you can see the highlighted red iodine molecules attached to two ring-like structures. Now, although this is an amine-based hormone, it does have steroid-like activity when it arrives at the target cells. Because of these organic ring-like structures, it does behave similar to a steroid. So they can be a little more lipid-soluble and access the interior of the cell a little more easily and find its receptors even on the DNA. So these effects can include glucose oxidation, how well you burn sugars from the meals you eat. We already saw how it can increase basal metabolic rate. We measured oxygen consumption and the weight of the animal to indicate their BMR, and also heat production. T3, T4 will help maintain blood pressure, regulate tissue growth, can influence sweat production, developing skeletal and nervous systems, and maturation and reproduction capabilities. So wide-ranging effects on target tissues, and that's why we have spent a lot of time working on T3, T4. Even though they're chemically similar, they need to be in the proper ratio. And when the thyroid gland has problems secreting T3, T4, goiters can occur, and also metabolic problems in the patient, which would have to be addressed maybe with a synthetic thyroid medication. If we turn the patient over and take a look at the dorsal side, notice in this diagram the trachea is no longer present. Now we see the esophagus, and we're looking at the back side of the thyroid gland, sort of like the butterfly wings that wrap around facing the dorsal or back side of the patient. So the parathyroid glands are peripheral, and I put big red circles around these 4 tiny pink parathyroid glands. And through the chief cells, they're going to produce parathyroid hormone, or PTH. PTH is very important in regulating calcium balance in the blood. And when your calcium levels become too low, either because you don't eat enough milk and cheese in a day to absorb calcium through the intestinal tract, which also requires vitamin D. And an easy way to gain vitamin D is to stimulate synthesis naturally by exposing your skin to the sun. So UV triggers vitamin D synthesis from the skin, and that will help absorption of calcium through the GI tract. When calcium levels are low, PTH is going to be released from the parathyroid glands, and it's going to target the bones, kidneys, intestines to try to balance out a low calcium level. Your teeth, your bones will be targeted, and osteoclasts will start breaking down bone and teeth in order to get your blood levels up. So usually women by about the age of 30 need 1.5 grams of calcium a day, and it's almost impossible to eat that much cheese and milk in a day. So you really do have to supplement calcium at some point if you want to avoid osteoporosis later in life or brittle bones. So once the calcium levels start rising in the bloodstream, that will start inhibiting PTH release, which again would be a classic negative feedback control loop to optimize calcium levels in the bloodstream. Now let's talk about the adrenal glands. We initially learned about adrenaline from our central nervous system unit, and when the brain perceives fear or danger, the sympathetic preganglionic neuron has a direct synapse to the middle or medullary layer of the adrenal gland. And that's shown here in the darker brown in the core. And that is a true neuro response. So the neuron is synapsing with the cells inside the adrenal medulla, and epinephrine is released, or adrenaline, in an 80-20 ratio. And it has a global mass activation effect. So intestines would be slowed, muscles, heart rate, eyes will be dilated, and the delivery of extra oxygen and blood for the fight or flight response. So that was the neural portion first introduced in Unit 2. I'd like to switch and emphasize the true glandular tissue since that is new material for Unit 3. And that takes us up to the adrenal cortex. There's a rectangular box highlighting the fluffy upper 2/3 of this organ. Notice how the adrenal glands look like little pyramid-shaped glands that sit on top of each kidney. And if we consider the different hormones that come from the stimulation of adrenocorticotrophic hormone, or ACTH, in the blue box over here on the right, that is the trophic hormone that is going to regulate the release of several important steroidal hormones from these different zones. So from histology, which is when we stain tissues and look at them under the microscope, we can see that there's clearly 3 different types of textures or layers within this fluffy glandular zone. And the first zone is glomerulosa, and it has kind of a blue staining effect. And then zona fasciculata is in the middle. And if you go down to zona reticularis, it has a slightly different texture and darker staining capabilities. And zona reticularis will sit right on top of the neuromedullary portion. So we're just talking about the 3 zones on the upper cortex, which is true glandular tissue, and the three distinct zones are responsible for aldosterone, glucocorticosteroids, and the sex hormones. So the different types of corticosteroids are produced in each of these three layers or zones and controlled by the trophic hormone ACTH. Zona glomerulosa is responsible for the mineral corticoids, or mineralocorticoids is what they're formally called, primarily aldosterone, and this helps regulate sodium reabsorption and potassium secretion by the kidneys. So you can think of aldosterone as being sort of an electrolyte hormonal balancer. Usually in times of low blood pressure or low blood volume, retaining salt will help you retain water, and that will keep your blood volume up and your pressure. We'll talk more about this in the circulatory system studies in Unit 4 coming up. Zona fasciculata produces glucocorticosteroids, or glucocorticoids for short. Primarily cortisol, which helps regulate the body's response to stress and raises blood sugar levels by inhibiting glucose uptake by the cells and stimulating gluconeogenesis. So under times of stress, we need extra energy to power our muscles and get us through times of stress. And then the bottom, zona reticularis, our last zone, the gonadocorticoids, mostly androgens, and androgens are responsible for the sex hormones that regulate reproductive function, as well as secondary gender characteristics, which we normally associate with being female or being male. So when the body has times of stress and it needs to resist this, keeping blood sugar levels even and relatively constant is a benefit of having steroid hormones around and maintaining blood volume, which I just mentioned. But cortisol can also provoke gluconeogenesis, so that will raise blood glucose levels. And gluconeogenesis, if you remember back from Unit 1, neo meaning new, genesis the creation of, means glucose from non-carbohydrate sources. So burning fats, using amino acids to raise the blood sugar levels to help out during times of stress. But if you were to be on medical steroids, where you are taking steroids supplemental for a medical reason, excessive levels do tend to depress cartilage and bone formation. We typically think of steroids as inhibiting inflammation, but that also means that we're slowing the healing process. Inflammation, the four cardinal signs, redness, swelling, heat, and pain, are things that we don't like. So steroid hormones can help cool the effects of inflammation, but know that inflammation is a normal part of the healing process. And when your body's giving you pain signals, that usually means you need to take it easy or stop doing that. High levels of corticosteroids can also depress the immune system and promote changes in cardiovascular, neural, and GI function, as well as reproductive function. So doctors typically prescribe these drugs at a certain period in the healing process because it will slow healing. So usually after maybe 3 to 4 weeks, your stitches come out, you might get some steroid medication to help with signs of inflammation at that point, but then the primary healing has already begun. Chronic stress and lack of sleep can induce high levels of cortisol that can cause a number of negative effects listed here. Atrophy of the hippocampus. The hippocampus and the amygdala were part of the limbic system that we studied in Unit 2, and those structures are involved in memory. So stress and lack of sleep obviously affects memory. Maybe you've been tired for an exam and didn't do as well as you thought because you were stressed out. It can also reduce the sensitivity of tissues to insulin, causing insulin resistance and a diabetic state where high blood sugars are circulating. It can also inhibit cranial nerve 10 and suppression of growth hormone, thyroid hormone, and gonadotropins, all of which we probably don't need. Okay, so if we switch to the pancreas now, again, another example of a dual functioning organ. Most of us think of the pancreas as part of the digestive system, and that would be correct. The exocrine function would be the gastric juices that are released near the duodenum, which is the bottom part or the distal portion of the stomach. But you may also think of the pancreas for secreting insulin, the hormone that comes around after a big sugary meal. Insulin will help with glucose absorption into the cells where glycolysis can occur and lower blood glucose levels. The histology involved with the pancreas is where we discover the production of insulin and its antagonist, glucagon. Glucagon is a hormone that's present when glucose is gone in times of starvation or when you have low blood sugar levels. So insulin and glucagon work in opposition with each other to maintain healthy blood glucose levels. And on histology, the islets of Langerhans are the structures that produce these hormones. The acinar cells produce the enzyme gastric juices used for digestion, which is the exocrine product. We want to focus on the endocrine products. So here on histology, this is back in chapter 6, figure 10, glucagon production. This is an amino acid polypeptide hormone, and it is going to raise glucose levels. So it's a hyperglycemic agent, unlike insulin, which is a hypoglycemic agent. It's going to lower blood glucose levels. So on this diagram, we can see that the alpha cells responsible for glucagon production are large and they take up a lot of the pink stain. And then if you look below the red circle, beta cells responsible for insulin production are much lighter. They don't take up as much of that dark stain and they're small. So there are two different cell types inside the islets of Langerhans that will help produce the endocrine products, insulin and glucagon. Here's a diagram of insulin. It's a relatively simple amino acid-based hormone. So this is a peptide. Peptides are short proteins, 50 amino acids or less. And if you look closely, you can almost read the sequence of amino acids in the beta chain shown in purple and the alpha chain shown in blue. These two peptide chains are held together by disulfide bonds, and this sort of curvy shape is what interacts with cells after a big meal. So the beta cells and the islets of Langerhans produce insulin, and it's going to function to lower blood glucose levels, enhance the transport of glucose across the plasma membrane and all body tissues, and it's going to counteract metabolic activity that would otherwise enhance blood glucose levels. And in this diagram, also probably not in your book, so you can get this study from the notes. I'm hoping that these notes are more helpful than just reading the book. Got some diagrams here that show the glucagon pathway in gray and the insulin pathway in purple. So both emanate from the pancreas. Both are going to target the liver. And remember that the liver is important for regulating blood glucose levels. So it needs to be stimulated to release glucose in times of need, times of starvation. And when we're in the well-fed state, it can store glycogen for in times of need. So the hyperglycemic effects of glucagon secreted from the islets of Langerhans, the alpha cells, is in contrast or is antagonistic to the hypoglycemic effects of insulin secreted by the beta cells. They work together to maintain healthy levels of blood sugar. So when you have high blood sugar, it's going to stimulate the purple insulin pathway. Insulin's going to target tissues and cells to absorb sugar and lower blood glucose levels. And the opposite is true when you're in starvation mode. You don't have very much blood sugar. You haven't eaten in 24 hours. That's going to stimulate the pancreas to secrete glucagon. And then the stored Glycogen can then be used as a source to raise blood glucose levels. If you need to go back and look at the structure of glycogen, please do so. That was introduced back in chapter 2. Okay, and finally the pineal gland. I think we tried to locate this in lab number 4. It's a posterior gland, very tiny, smaller than the size of a pea, and it may have been dissected out already when you had your sheep brain available, and it's also difficult to see on the brain models that we had. The pineal gland is responsible for the secretion of melatonin, and you can see the molecular structure of melatonin here. Again, those ring-like structures will make it a little bit fat soluble, so it may have some steroidal-like interactions with its target cells. Melatonin is secreted in response to low light. So the suprachiasmatic nucleus in the hypothalamus is part of our regulatory system that sets our circadian rhythm. So light and dark stimulation are going to control the release of melatonin. Melatonin is secreted only at night, and it helps you feel sleepy. Nowadays, you can supplement melatonin by little gummies, and they're reported to help you sleep. So light and dark information is going to reach the brain through cranial nerve 2, and you can follow the pathway of stimulation at night and inhibition of this hormone during the day through the eyes. And melatonin is going to peak between midnight and 8 a.m., help you feel sleepy. So this hormone helps us set our circadian rhythms. And another gland, a little more minor, is the sweetbread or the thymus. And you can see that it is nestled between the lungs, and it's fairly large. It would be mediastinum. The heart is covered by the pericardium, but you can see the thymus nestled between the lungs here, right along the sternum. And this gland is really responsible for the development of your immune system. So T cells are taught what to look for by the thymus. So hormones that are essential for T cell development, which would be T killer cells and cells of our immune system that will go and target infected cells, including viruses, parasites, things like that, as part of the adaptive immune system. So once we're exposed to an antigen, we can develop T cells or B cells. We'll be looking at B cells, which are lymphocytes from the spleen that will secrete antibodies, and they are in direct response to foreign invaders. So our immune system is very pliable and adaptable based on the different disease-forming molecules we may have been exposed to during our lifetime. This type of activity usually becomes a little less active after adulthood. Many children get sick. We try to vaccinate when possible, but children just don't have the development of the immune system like adults do. We've been exposed to a lot more antigens in our lifetime than would a newborn baby who has no immune system other than the passive immunity that it would get from breastfeeding on its mother. Okay, so let's come back. Now that we've covered most of the major glands, I'll talk about the secondaries at the end of this section, but let's talk about the actions and interactions of all these hormones we just listed and try to understand better what happens at the target once they arrive from the bloodstream. This is an area where you also need to make a list or flashcards or something to help you organize these different chemical classifications. Is it a steroid? Is it a glycoprotein? Is it an amine-based hormone? So take this information and use it to categorize what is a steroid. Steroids are all lipid-based because they form from cholesterol. You must have cholesterol in your diet. I know it gets a bad rap in the media, but cholesterol is essential for cell membranes and the production of steroid hormones. So we talked about gonadal and adrenocortical steroid hormones, testosterone, estrogen, progesterone, and the cortisol family. Those would all be under the category of steroids. Now, amine hormones are derived from amino acids, mainly tyrosine and tryptophan. There's 20 amino acids total, but tyrosine and tryptophan are the ones that are used in hormonal production. And those would be epinephrine, norepi, thyroxine, T3, T4, and melatonin, which we just discussed, being secreted from the pineal gland. Polypeptides, remember those are short proteins. Peptides are usually 50 or fewer amino acids in a chain. And those would be vasopressin, or antidiuretic hormone, growth hormone, insulin, oxytocin, glucagon, adrenocorticotropic hormone, and parathyroid hormone would all fall under this category. And I did show you what insulin looked like, and it was by far one of the more simple structures we've looked at. And then glycoproteins include luteinizing hormone, follicle stimulating hormone, and thyroid stimulating hormone. Hormone actions produce major changes that I already discussed at the beginning of this lecture. If you're a target cell, hormones can cause big changes, metabolically, structurally, and secretory. Even mitotic cell division can be triggered by some of these hormones. So we are going to focus on the activation pathways, but just know that a target cell has to have a specific receptor in order to respond to a signal. Otherwise, it would be deaf to that signal and would not be able to respond. And we also need a way of inactivating hormones. So that's why we looked at the negative feedback control systems that help maintain hormone balance. Now, again, another review of how steroid-like hormones, including the thyroid hormones, can easily access the cytoplasm of a cell because of their solubility. All steroid hormones being cholesterol-based and thyroid hormones with those ring-like structures are going to make them act like fat-soluble molecules. So when they arrive at their target cells, we don't see a receptor being presented on the surface facing the ECF compartment. The solubility, like dissolves like, means that steroid-like hormones can use the fatty acid chains in the plasma membrane to dissolve directly across the barrier and have access to the inside of the cell. So they may have the receptors in the cytoplasm, and also because the nucleus is a plasma membrane-bound organelle, these powerful chemical ligands can access the nucleus and affect protein synthesis, mitosis, some of those effects that we already listed. So with these types of fat-soluble hormones, they're going to find the receptors inside the cell. And this can cause really major effects at the cellular level. So the target cell activation depends on factors like blood concentration levels, relative numbers of receptors on the target cell, and the affinity, which is the attractive forces between the receptor and its hormone. Remember, we're not talking about covalent bonds here. We are talking about some of the weaker bonds like hydrogen-hydrogen interaction and van der Waals. So these can be easily used as kind of an on and an off when you have some of the weaker bonds. Target cell activation can be upregulation, so target cells can form more receptors in response to the hormone, or downregulation where they can lose receptors in response to the lack of hormone, and tissues can respond to many different hormones. And that means that if you have multiple hormones, 2 or more around at the same time, they can tend to have a synergistic effect on their target cell. So if they work together, they end up producing an exaggerated or stronger effect in the target cell when you have two or more hormones working on the same target tissue. The opposite would be true if one of those hormones is sort of inhibitory to the effect of the other, and that would be an antagonistic type of situation. So hormone blood concentration levels are controlled by negative feedback systems. Variations are limited and occur only within a narrow range. Hormones are synthesized and released in response to three main types of stimulation. Again, doing a flashcard, trying to associate the next three slides I'm going to share with you with either humoral, neural, or hormonal stimuli. Two out of the three we have already had practice with. And if you go back to your pre-lab from lab number 6, you guys listed these for me. And it might be a good time to review what you might have written down during the lab to get the 5 points for participation. You may want to look at this in a little more detail because in lecture, we do have more details that we're responsible for than we do in the laboratory where you're just doing a procedure. So humoral is the pathway that you're probably least familiar with. Humor is the Latin term for body fluids. And remember when we looked at the special senses and we studied the eyes, we had the aqueous humor and the vitreous humor, and those fluids kept the eyeball in its normal shape. Humor, in this case, would be the fluid portion of blood. So secretion of hormones in direct response to changing blood levels would be a humoral stimulation pathway. We have studied neural stimulation for 6 weeks now. So nerve fibers that are sending action potentials can stimulate hormone release. We saw that with the hypothalamic pituitary axis. And then hormonal stimuli. We have talked about trophic hormones, hormones that control the release of other hormones. So we're going to go back now and look at these pathways so you have more of a visual. And some of these examples will be given to you on quizzes and exams. So try to remember that neural control is going to be an action potential causing the release of some neurotransmitter that then stimulates a hormone. So nerve fibers extending off of the central nervous system, targeting, now we're looking at the adrenal gland, but the middle medulla, the medullary portion is neural, and that is responsible for dumping adrenaline into the bloodstream. Epi-norepi in an 80-20 ratio is going to flood the bloodstream and target every tissue in the body. That's called a mass activation, and the body's going to respond with the sympathetic nervous response or fight or flight. Epi-norepi is under the category of catecholamines. Okay, so the adrenal cortex will be able to respond with the release of cortisol. Remember, steroids help us out during times of stress. Steroids just aren't great long term. So there have been several examples of the neural pathway, and the one you should expect most is probably the hypothalamic pituitary connection to the posterior pituitary, which is more germane to this chapter. Humoral. Okay, so circulating blood levels. In this example, we're looking at the parathyroid hormone that's released from the parathyroid glands on that posterior view or the dorsal side of the patient, where the butterfly wings of the thyroid gland have the parathyroid glands embedded there, and they are responsible for the release of parathyroid hormone, which regulates calcium levels. So whenever PTH is around, it's going to target bones and teeth, the GI tract, anywhere it can to try to cause blood calcium levels to rise. Once they rise, the blood levels feeding this gland will tell it, hey, we have enough calcium, and the PTH stimulus will be removed. Again, classic negative feedback to control circulating blood levels. We also saw a long and short negative feedback loop on the hypothalamic pituitary thyroid connection, which we practiced in lab. So all of these hormones are going to be regulated through negative feedback control mechanisms. Hormonal stimulation, so trophic hormones that control the release of other hormones. So again, the hypothalamic pituitary connection, the anterior pituitary is true glandular tissue. So thyroid releasing hormone to thyroid stimulating hormone to the thyroid gland to secrete T3, T4. Stimulation of ACTH targets the adrenal cortex for the steroidal hormones that we just discussed. Growth hormone, gonadal trophic hormones that will secrete LH and FSH from the gonads. This is a multi-step pathway, and I would expect some or all three of these on exam questions coming up at the end of this unit. So to finish up, let's just talk about some miscellaneous glands and their hormones that maybe normally you wouldn't think of these organs as being hormone producers, such as the heart. When we finish with skeletal muscle here in Unit 3, we will apply what we learned to skeletal muscle structure and function and start talking about the heart. The lab experiments are already starting with the heart, and this is typically thought of as a pump. So it drives circulation. It pumps your blood. This is a 4-chambered hollow organ, and striated muscle is responsible for the contraction and relaxation of the heart. But the upper chambers, which act as receivers of blood, are able to respond to blood volume changes. And atrionatriuretic peptide is going to be released from the 2 upper chambers, the atria, and it will communicate with the kidneys to alter blood sodium concentration that will affect blood pressure and blood volume. Also, the GI tract, there are hormones that will act locally. The kidneys, you think of the kidneys as filtering your blood and producing urine. They actually secrete the hormone erythropoietin, and that targets the long bones where bone marrow is located in the center. And bone marrow is where red blood cells are formed and some of your immune cells. And red blood cells have about a 3-month lifespan, so they don't last forever. The spleen will recycle the iron and the hemoglobin, get rid of things that might be toxic, and new red blood cells will be generated through the bone marrow under the control of erythropoietin. I already mentioned vitamin D synthesis from the skin when it's exposed to UV. And finally, adipose tissue. Adipose tissue releases the hormone leptin, which tells the brain you're satisfied or you're full, and it can also stimulate use of more energy because you're in the fed state. So patients with severe obesity tend to have a lot of steroid hormone imbalances because those hormones are fat soluble, and having too much adipose tissue forms like this storage vessel for hormones that are fat soluble, and it can also disrupt the release of leptin, which tells your brain, stop eating, I'm full. So this is an active area of research to see if the supplement of this hormone can help with appetite control. So you may be hearing more about this in real life as you progress forward.